Life After Interphase with Natasha and Anika, Meiosis edition.
Before we begin, we wanted to explain the overall purpose of meiosis and explain why meiosis occurs in the first place. The overall purpose of meiosis is to produce cells that not only possess half the chromosome number of the original parent cell, but that are also genetically different from one another in the original cell. This type of division is required in order to produce egg and sperm cells for sexual reproduction. Meiosis is an important process that can enable genetic diversity and aids the repair of genetic defects through cell division and replication. Now let's learn more about meiosis. First, the cell exits interphase leaving the G2 checkpoint. The G2 checkpoint is represented by the door labeled G2, and the spindle fibers are seen being represented by the yellow glow stick. At this point in meiosis, the chromosomes are not condensed or conjoined. The G2 checkpoint ensures that the cell is not entering meiosis with the damaged DNA.
After interphase, the cell enters prophase 1. This is the first stage of meiosis. In this phase, the chromosomes condense and the nuclear membrane dissolves. Chromosomes cross over and bind together, creating tetrads. Here, we have shown the nuclear membrane, which is represented by the green glow sticks, formed in a circular form, and the chromosomes are represented by the pink and blue glow sticks. This is shown by the pink and blue glow sticks mixing together. Spindle fibers also emerge from the centromeres, which are represented by the yellow string.
Step 2 meiosis is metaphase 1. In metaphase 1, the spindle apparatus sends out spindle fibers to attach to the chromosomes, and since they are lined up side by side for crossing over, they are tightly held together. During this phase, the chromosomes cross over, which is represented by the changing colors on the chromosomes, or in this case, it's represented by the crossing over of the glow sticks.
In anaphase 1, homologous chromosomes are pulled apart and moved towards opposite ends of the cell. However, the sister chromatids stay in place. During anaphase 1, reduction occurs with 23 chromosomes moving to each pole.
Finally, to complete the first part of meiosis, the cell undergoes telophase 1. In telophase 1, the cells are now at opposite ends of the cell and form two distinct chromosome clusters. During this phase, nuclear division begins and the parent cell is divided in half. Each daughter cell will have 23 chromosomes. Each chromosome consists of two sister chromatids.
After telophase, the cell is divided into two daughter cells by the division of the cytoplasm of the original cell, which is called cytokinesis. This now allows the cell to move on to meiosis 2. Meiosis 2 is the second phase of cell division with the purpose of separating sister chromatids. The main difference between meiosis 1 and meiosis 2 is that in meiosis 1, it is the process of separating homologous chromosomes, but in meiosis 2, it is the process of separating sister chromatids.
Meiosis 2 is the second division of meiosis. It begins with prophase 2. During prophase 2, the chromosomes start to recondense and spindle fibers form once again. The spindle fibers from each side of the cell attach to the other sister chromatids of the chromosome and move the chromosomes until they are lined up at the spindle
The next
equator.
step of meiosis 2 is metaphase 2. During metaphase 2, each of the 23 chromosomes line up along the center of the cell at the metaphase plate.
After metaphase 2 is anaphase 2. During this phase of meiosis, the centromere splits, which frees the sister chromatids. The spindle fibers start to shorten, allowing the newly separated sister chromatids to move to opposite sides of the cell. The sister chromatids are not genetically identical because of crossing over.
The last official phase of meiosis is telophase 2. During telophase 2, the chromosomes begin to decondense and the nuclear membrane reforms. The sister chromatids are represented by the individual green, pink, blue, and orange glow sticks. Then comes cell division, which is done through the process of cytokinesis, and as a result, 4 haploid cells are formed.
Thank you for watching and listening to the Life After Interphase with Natasha and Anika Meiosis edition.